Afternoon, everyone. I'd like to introduce you to Vor Bio. We are a cell therapy company, really trying to do things a little differently. I will point you to our disclaimer, which you'll be able to read at your leisure on our website. So what is Vor? So Vor, we're trying to cure cancer, and we don't use that word lightly. Cancer is terrible, but particularly blood cancers. We've had a lot of revolution in the last 5 years, but particularly in B-cell cancers where these cells are disposable. You don't really need them to survive. However, with other kinds of cancers such as bone marrow cancers like AML, this is a cancer which takes over your bone marrow and ends up squeezing the life out of you, and unfortunately killing cancer cells in your bone marrow. They look a lot like your healthy bone marrow cells. And so we take quite a unique approach here. Firstly, we have a sword. So that sword is a CAR T therapy. This CAR T is a little different, though, to other CAR Ts you are probably familiar with, in that we actually use cells derived from healthy donors to make these CAR T cells. That gives us a special advantage in terms of the phenotype of cells that we can produce for the CAR T. Secondly, and maybe more importantly, we actually have a shielding strategy as well. And that shielding strategy provides for a shielded treatment-resistant stem cell therapy for these patients with AML. And these stem cells and their progeny, the whole blood system of the patient, we hope will be immune to targeted therapies such as our sword, VCAR33 We are proud that we actually have in-house manufacturing capability, but we also use CDMOs, and we really try to harness the strengths of both. So just to think a little bit about AML. So as I mentioned, AML is a blood cancer, but it's derived from bone marrow. It's very common. It's actually the most common acute leukemia in adults, and is a terrible disease. In fact, the standard of care in AML is a stem cell transplant. So what is a stem cell transplant? This was actually the first cell therapy ever produced, where you can actually poison the bone marrow to the extent that you kill hopefully all the cells in the bone marrow, but of course you have to replace it with something. And you can't replace it with cells from the patient because that has cancer, so you need to replace it with stem cells from healthy donors. You now know that you can HLA match donors to replace the bone marrow from patients, and that's exactly what a stem cell transplant is. Unfortunately, you can see there on the right, patients do very poorly. Even despite the poisons they get to replace your bone marrow, you can see patients who are at high risk of relapse. The top two curves are roughly 70% likely to relapse within 12 months. That represents actually 50% of all patients undergoing a stem cell transplant. These are exactly the same patients that we're targeting in our clinical trial. And so we have these three approaches. Let me talk to you firstly about the sword, which is VCAR33 So again, when you think about CAR Ts, we know a lot about the left side. We know that autologous cells, so taking cells from a patient, inserting a CAR construct with lentivirus or other means, is very effective, and particularly in B-cell cancers or plasma cell cancers. Unfortunately, in diseases like AML, because of the repeated therapies these patients receive, these cells may not necessarily be of the healthiest phenotype, and that can lead to manufacturing failures. Similarly, allogeneic off-the-shelf approaches have had a real revolution in the last few years, but unfortunately have not met up with the promise of autologous approaches, really because it's hard to hide these cells from the immune system. Our immune systems are very smart. They've been trained billions of years to figure out what is native and what is not. And cells from a different source tend to get eliminated very quickly, perhaps more so quickly that they cannot cause the treatment effect that you're targeting. So instead, we're doing something different to both of these. What we're doing is we are treating patients who have relapsed after their first stem cell transplant, and we're going back to their original donors and sourcing cells from these donors. So why is that interesting? Well, firstly, these donors are healthy. They've never had chemotherapy, so of course their cells are very healthy in phenotype. But what's also intriguing is that these donors their cells were responsible for repopulating the blood system in these patients. And so these cells are inherently matched to the patient. And so we're taking advantage of a unique biological circumstance where these cells are exactly matched, but they do not come from the patient themselves, really being ideal starting materials for CAR T. And this shows the kind of phenotype of cells that we're able to generate. So it's a very rapid manufacturing process, around 7 days. And you can see from the blue bars, these are exactly the types of cells that we're targeting. These are stem-like, long-lived cells capable of in vivo expansion and persistence. This is what we are doing is we're targeting a protein called CD33. So CD33 is almost the perfect target in AML. You can see on the left that cells almost ubiquitously express CD33. And then on the right, you can see that antigen density is high, such that there's a lot of targets per cell. However, one of the difficulties of CD33 is that it is expressed on healthy cells as well, but we'll get to how we solve that solution with our shielded approach. What we're doing in the clinic right now with the CAR T is we are treating patients with MRD positive or relapsed refractory AML. So they enrolled into the study. Unfortunately, there's a lot of these kinds of patients. We then approach the healthy donor, we harvest cells from that donor, and manufacture VCAR33. So they receive that therapy after lymphodepletion, and then we are able to enroll patients both with MRD positive disease as well as those in true relapse, and we will follow these patients up over time. This is the trial that's actually ongoing today in around 7 or 8 sites around the United States. And we're expecting to read out data later on this year, which we're really excited about. So let's talk a bit about the shielded transplant approach, trem-cel. So with trem-cel, this is a different trial design. What we're doing is we're taking patients who are eligible for a stem cell transplant. However, these patients have been categorized as having a high risk of relapse after the transplant. That could be because of MRD positive disease, it could be because of certain genetic markers, cytogenetics. And these patients enter the study. Of course, we work with healthy donors who are HLA matched to these patients to donate stem cells, and then we manufacture trem-cel. We manufacture this by using CRISPR-Cas9 gene engineering. We remove the expression of CD33. Now, those cells are infused as a substitute of the regular stem cell transplant. And then we wait for engraftment, which is the ability for these cells to reconstitute the blood of the patients. And we also administer Mylotarg typically being very toxic after a stem cell transplant. So we've had the fortune of treating several patients with this approach already. We can answer some really fundamental questions about trem-cel and CD33 biology through this clinical trial. For instance, firstly, we can put this through a robust manufacturing process where we can effectively knock out the expression of CD33 in, as you see, roughly 80 to 90% of cells that we process. Not only is that knockout almost complete, but also these cells essentially replace the entire bone marrow of these patients. You can see the chimerism at the bottom is essentially perfect. The cells in the patient's bone marrow are almost entirely made up of healthy donor cells. That's exactly what you want. Now, before our clinical trial, no one had done this kind of experiment where you knock out CD33, knock out a protein that is highly expressed on myeloid cells, and expect these myeloid cells to function normally. And what you see on the right is that the engraftment of these cells, as depicted by reconstitution of neutrophils, is essentially perfect. We're expecting engraftment to be around 11 days, according to a robust clinical trial, and our patients have engrafted at 11 days or even earlier. So I think we've shown even in the first 8 patients that we can robustly reconstitute the immune system of these patients even without these cells expressing CD33. Now, we are dosing Mylotarg to these patients. Mylotarg is an antibody-drug conjugate that targets CD33. It is typically very myelotoxic. You can see that on the left side. After dosing, typically these patients slip into grade 4 neutropenia or thrombocytopenia, and there's nothing you can do about it. Our patients, you can see 3 patients we've read out later on already, have very robust cell counts. So both neutrophils and platelets do not seem to waver much at all on the basis of that these cells no longer express CD33. So it really does show that we can shield the blood system of these patients by using Trem-cel that does not express CD33. Now, how do we know Mylotarg is working? Well, we certainly know it through pharmacokinetics, and we published that data. But what we can also see in these patients where we've dosed Mylotarg is that there's an enrichment of CD33, such that the patient's blood and immune cells are no longer expressing CD33 and progressively getting wiped out through the use of Mylotarg. So we certainly know Mylotarg is working and potentially keeping their relapse at bay. So, how do these combine? They combine on what we call a treatment system. So, standard of care, again, patients relapse very commonly. What we want to do is enable what is described at the bottom, where patients receive a shielded stem cell transplant, and they can also receive a CAR T, a sword that's normally very toxic, but in our hands, hopefully, is not toxic because of the shielded graft that they've already received. And so you can imagine an environment where this shielded graft protects the blood system with this potent sword sourcing anything to do with cancer. And this is how we're manufacturing our product right now. We are creating Trem-cel, which is shown on the top row, where we efficiently delete out CD33 using CRISPR-Cas9. And we have the potential of making the CAR T from exactly the same source material as the stem cells. Again, this is healthy donor-derived, so able to create a very potent CAR T as well as a shielded transplant from exactly the same source material. So let's step back and think a little bit about what Vor is doing. We harness both contract manufacturing as well as in-house manufacturing. Our in-house facility is modest. It's 4 clean rooms that we actually assembled in Cambridge, Massachusetts. It's really built as a way to supply first-in-human clinical trials. And that facility, in fact, is making VCAR33 today. We also use a contract manufacturer. We use several contract manufacturers for critical raw materials, but also we even use one for cell therapy manufacturing. In fact, the CEO of this facility is sitting in our audience, Louisa from C3I. We're very proud that C3I is a key partner for us manufacturing Trem-cel. C3I takes our stem cells that are shipped to them, they process these cells to remove CD33 expression, and then they ship it throughout our clinical trial sites throughout the US and Canada. And they're able to provide us tremendous flexibility, and we have complete confidence and trust in them as a CDMO just as much as we do as our in-house facility. So just stepping back and thinking about what's next for Vor. This year is a really important year because what we're doing is that we've already shown proof of principle, which we characterize as can we delete out CD33 safely, and can we shield the blood system from what would normally be a toxic therapy? We think the answer to those is yes. But what we want to be doing is moving to the right side. We want to show that not only can we shield the blood system, but we can actually reduce disease burden and change the trajectory of this disease in patients. And so what we're also thinking about is how do we push it forward from here? How do we think about pivotal trials, even moving into commercialization? And so what we want to do is marry, again, Trem-cel, combine it with the sword, the power of VCAR33, such that we can delay relapse and we can truly aim for cures for these patients in need. I want to thank you very much for listening.